Chapter 7 of our textbook poses a very interesting question. Are you only as smart as your genes? And so in the introduction to this chapter, the textbook discusses a place near Washington known as Fairfax Cryobank. Now you may have never heard of this before, but it's a sperm bank. Inside of a sperm bank, obviously there are sperm cells that are being stored, and these particular sperm cells are what this company does is women can come, they can purchase the sperm in hopes that they could get pregnant and conceive a child. What's interesting about this sperm bank is that donors that donate their sperm are categorized into 3 different classes, and their sperm is priced accordingly. What's even more so interesting is the donors that the sperm come from. If they fall into the graduate category, which means that this particular man has a degree in either medicine, in law, or some type of academia, their sperm is priced 30% more expensive than those that don't have a graduate degree. So the question would be then, why are some women willing to pay significantly more money for sperm that come from a donor that has an advanced degree? Is intelligence about genes? Meaning, is intelligence something, excuse me, that you can inherit? Or is intelligence a function of the environment in which you were actually raised? And so in order to answer those questions and to understand this a little bit more in detail, we need to learn a few things about inheritance. And so in this part A of Chapter 7, we're going to look at some vocabulary terms, genes, chromosomes, and alleles. We're going to define the life cycle. And then we're going to explain why cells that contain identical genetic information actually look different. And then going back over a couple processes that we talked about in the previous chapter known as segregation and independent assortment. So to begin with, let's think about families. You know, if you have siblings and you share the same parents, one of the things that you probably have noticed, unless you're identical twins, is that you all look a little different. Now, you may have some characteristics that are similar to your mother or to your father, and you and your siblings may share some characteristics that look similar, but each child in your family is unique. And the reason that we're unique is because we're not just an average of our parents. It's not about a blending of the two. It's actually what we call a reshuffling of traits. You notice if you look in some families, one child may be similar to mother in eye color and face shape, while another child is similar to mother in height and hair color. And so think about your own family, and think about your siblings and your parents, and think about the characteristics that you share with them. And again, you'll see that every one of you is unique. Now, why is that? Well, let's start talking about something known as the life cycle. Now, we know the human life cycle, it all begins with meiosis, and meiosis being a process we talked about in Chapter 6. Remember, the purpose of meiosis is to produce sex cells or gametes, sperm cells and egg cells. And the whole purpose in making sperm cells and egg cells is that so that those two things can fuse together during fertilization, and they form that zygote. Remember, we talked about that single-celled zygote that we all started out as. And through a process called mitosis, that zygote splits into 2 cells, into 4 cells, and before you know it, it has divided enough times to become an actual embryo. And then over time, the embryo grows into a fetus. Fetus grows into a full-term infant, and throughout the lifetime becomes an adult. But what's interesting to note, and what's really, really important to remember here, is that all of the traits about you, whether you're a teenager or whether you're an adult, all the traits about you were influenced by the information that was contained in that single-celled zygote. And where did that zygote come from? Remember, it came from the fusion of sperm cell and egg cell. It came from the fusion of your parents. So again, children are going to share similarities with their parents because children are inheriting their information from the parents' genes. Now, remember, gene here is not the denim that you're wearing. Genes here are segments of DNA, and those segments of DNA contain information that help your cells make proteins. And then those proteins are what carry out expression. So remember, genes are found on chromosomes. And depending on if you're a prokaryote or eukaryote, remember chromosome structure is different. So for prokaryotes, remember that don't have a nucleus, their chromosomes are single and they're circular. They're loops. For eukaryotes, chromosomes are linear. Remember, our DNA is found in that double helix, that spiral staircase, and it condenses into linear-shaped chromosomes. And remember, for specifically human cells, our body cells have 46 chromosomes. So a couple of statements to ponder as we're leading into this is that every cell in your body, nearly every cell in your body, contains the exact genetic information. Same number of chromosomes, same number of genes. So every single cell in your body has 46 chromosomes, and each of those chromosomes contains genes. Now what's interesting though is even though our cells are containing the same chromosomes and they're containing the same genes, how the genes are read is different from one cell to the other. For example, take your eye cells, cells that make your eye, and your pancreas cells. Both of these cells, again, containing the same chromosomes, containing the same genes. And so what's interesting is that both eye cells and pancreatic cells, they contain genes and instructions for building a protein known as rhodopsin. Now you may not know much about rhodopsin, but rhodopsin is actually a protein that helps you to detect light. We think about it as far as eye cells or pancreas cells are concerned, which one would need to be able to detect light? Obviously your eye cells would. So what your eye cells do is they take the genes that carry the instructions for building rhodopsin and it uses them. Pancreas cells, on the other hand, do not. So they have that ability to turn those genes off. Another example, eye cells and tongue cells. Eye cells carry the instructions for building a protein called transducin. Transducin is helping to translate light. Tongue cells have instructions for building transducin, but what's interesting is that transducin in your tongue has a different function. It actually helps you to detect the taste of bitter. So the important factor that we want you to understand here is that proteins coded for by our genes may serve two or more different functions depending on what cell they're in. It's kind of like words building sentences. You think about it, how many words are there? There are billions of words, right? And even more different types of sentences that we can build with just those same words. Why? Or how? Because we take those words and we use them in different combinations. And as we use them in different combinations, even though we may be using the same words, we can build different sentences. That's the same way that our cells work, is that even though our cells contain the same genes, as we put those genes in different combinations, turn some on, turn some off, we're able to build different proteins. So what we want you to, again, using these same terms, is take chromosomes and kind of think of them as being equivalent to pages of a book. So if chromosomes are pages of a book, then the genes on those chromosomes would be equivalent to words on a page. So let's just say that this book that we're building here is an instruction manual for building a human. So again, the chromosomes will be the pages, the genes will be the words on those pages. Now we already know that in order for our cells to reproduce, our cells to divide, the DNA and the chromosomes have to be copied. But keep in mind that when we copy chromosomes, we don't photocopy them. It's not like you put them through a copy machine and you press go. When chromosomes are copied, they're actually rewritten. And so because they're rewritten, sometimes mistakes occur in copying the DNA and in copying the chromosomes. And anytime these mistakes occur, they lead to mutations. And these mutations produce different versions of genes that we call alleles. So by definition, an allele is an alternate form of gene. So look at the bottom of the screen. Again, if we're saying that the chromosomes are the pages and the genes are the word, look what happens sometimes with alleles. See, in this particular scenario, here's the original copy. Here's the copy that comes from it. So the normal allele, gray, is spelled G-R-E-Y. But in this case, the mutation that occurs leads to a different spelling of the word G-R-A-Y. Now, in this particular mutation, both alleles have the same meaning. But look at this one. A different spelling here, you have strong or string. Now, even though it's just one letter off, these are two totally different words, have two totally different meanings. Or in some cases, a mutation can cause the mutant allele to have no meaning at all. So how do we see these alleles play out in humans specifically? Well, let's say, for example, you're dealing with the gene hair color. Alleles that we might see because for the gene hair color would be different versions of hair color. So you could see a brown hair allele or a red hair allele. If the gene were eye color, you could see brown eyes or blue eyes. If the gene were blood type, you could see type A blood or type B blood. If the gene were skin color, you could have white skin or black skin. So these alleles that occur give us different variations of a gene. And so this is how you can see in a family. We'll talk a little bit later on about dominant and recessive, but you can see in a family different hair colors show through or different eye colors show through, different skin colors because of the different variations or the different alleles that may be present for a gene. So keep in mind that both parents are contributing to these genetic instructions, but they're not contributing their entire manual. Remember, we said you're not just an average of the two. So they're not going to contribute their entire manual. We also learned in Chapter 6 that because of meiosis, producing those sperm cells and egg cells, the chromosome number is going to be reduced by half. So it produces those haploid egg and those haploid sperm cells. But keep this in mind, even though you're only transmitting half of your genetic information to your gametes, each parent is actually giving a complete copy of their instruction manual. Because remember, a human body cell has 46 chromosomes. Those 46 chromosomes are really 23 pairs. So each cell, by having 23 pairs, it has two versions of each page. One version that came from your mother, one version that came from your father, actually giving you two copies of the manual. And so then what you actually send on to your sperm cells or send on to your egg cell, even though it's half the number of chromosomes, it's really a full copy or a full version of that manual. We call these homologous pairs. Remember we talked about this term in Chapter 6. Is where you have equivalent chromosomes that are not identical. One's coming from the mother, one's coming from the father, but they're full copies. So it's kind of like having a 23-page copy from mother, 23-page copy from father, and so when you put those 23 pairs together, you make a full homologous pair. But now keep in mind, we mentioned at the beginning of this section that siblings are unique unless they're identical twins. And the reason they're unique is because the parents do not give all of their offspring exactly the same set of alleles. Remember genetic diversity? We talked about this in the last chapter. Genetic diversity occurs because of a couple of different reasons. One, random alignment. Remember during metaphase 1, when the homologous pairs go to the middle or the equator, random alignment says that they don't line up the same order every single time. They can line up in any kind of random order, and so because of that, when the homologous pairs separate, it increases a variety of offspring. Not only random alignment, but the segregation, when those homologous pairs do separate, it increases the chances that each gamete is going to get a different copy, a different version of the manual. Look at this one for example. Bottom of the screen. One possible sperm cell that could be produced, page 3 received a blood group gene from dad, page 9 received eye color genes from mom. But an alternate version of a sperm cell, page 3, received the blood group from mom instead, and the eye color from dad. So every time a sperm cell is produced, it's going to be genetically different than the others because they're not always going to receive the same pages from the same parent, just like they're not going to receive the same alleles from each parent. So there's about 8 million different combinations of chromosomes that are possible. So when you take 8 million different combinations for a sperm cell and 8 million different combinations for an egg cell, you get 64 trillion possibilities for random fertilization. So that goes on to tell us that your parents could continue having child after child after child, and unless they have identical twins, every single sibling is going to be genetically different, even though they're coming from the same parents. Because genetic diversity says that, again, even though they're both contributing to your instruction manual, they're contributing different alleles. And so because of that, you get a variety of offspring. Now again, we keep mentioning that unless you are identical twins. So really quickly, we did just want to establish what twins were. So you have monozygotic and you have dizygotic. Monozygotic twins are identical, and you'll see why. In this case, sperm fertilizes an egg to form a zygote. That zygote becomes an embryo, and then the embryo splits. And these two embryos are identical, which produce 100% genetically identical twins. In dizygotic twins, they're only 50% identical, because in this case, you don't have an embryo that splits, you actually had two separate embryos. This was where the female produced two eggs, both of which were fertilized, both of which became zygotes and embryos, and so you get twins that are no more similar than siblings, because they did not come from the same embryo.